Yes, I got it. Right. All right, we can get started. In the future, I want you, if you're a student, to remember your student ID number. And it doesn't really matter for the quiz, but when we get to the midterm exam, and there is an option only write in your student ID number and do not write in your name. So that I want to make sure that when we grade the lecture, your answers, we're not being biased by who you are, okay? So making sure to write your own number in, or at least write down a note that which number you wrote in, and so we can trace back. Well, so now today is actually the 5th lecture of this semester, and I just look at the curriculum that we have only 2 more lectures, and then we have the fun midterm, and I hope you guys will be ready by that time. Okay, so we are going to continue to talk about DNA sequencing this time. This is the 2nd lecture of that. And our topic today will not be the general introduction, and instead we're going to into a lot in details about bioinformatics and the statistical topics in this. So, let me see. All right. So again, I want to refresh your memory a little bit what we talked about last time. So the first part, we talk about a different type of genetic variants. That will include the SNPs and single nucleotide polymorphism. Remember, we talk about, when we talk about P, this polymorphism, usually the definition is you need to have a certain percentage of the population have it. So if that's a variant only yourself has it, that doesn't count. But actually, it really doesn't matter, but just be careful when people talking about this terminology. And we mentioned the transition-transversion ratio, and we will continue, we'll reinforce your memory today in the later part of the lecture. And the second part will be the short insertion deletions. And then there are longer ones, including the repetitive elements that will be the microsatellites, mini-satellites, repeated genes, and also different type of larger structure variations, like large insertion deletions, duplications, inversions, copy number variation, and so on. So we are going to talk about the different type of strategies for the informatics analysis towards the end of the lecture about larger structure variations. And we also talk about in terms of the biological question we want to answer, and because of the technology involvement, initially we used microsatellites and then pretty loose SNPs and then much denser SNPs arrays. And the analysis will be either family-based, the linkage analysis, or just a simple the association analysis. Look at the association of different genetic markers with the phenotype of individual patients. And that usually is called the GWAS, genome-wide association studies. For this part, I want you to really be very clear what are the assumptions of the GWAS. It's actually a common disease, common variance, this particular assumption. And that is one of the reasons that when we do GWAS, usually initially people use the GWAS arrays, the genotyping arrays. And every time they put a probe on the array, usually it's over more than 5% of the population has that variant. So which means GWAS only, most targeted on the common variants. And then we talk about this spectrum. So there is whether it's common or rare, which is the X part. And for the Y part, it will be the effect size, can be large or small. So if something is very common, at the same time, the effect size is very large, usually that is not associated with a disease phenotype. It could be something, other things. But if it's looking in this area, not many people have it, and then the effect size is not very big, nobody cares. And but this spectrum is important. The first type is it's pretty common, but the effect size is smaller. And that is where the GWAS is really focusing on. And the other spectrum of this will be, it's very rare, but at the same time, if you have it, you have a much larger risk comparing to the one who doesn't have it and to get into the disease. This is also very important. And in the past, when we try to study this, GWAS won't work because GWAS doesn't really give you the rare variants. So it's only common variants. But this part, usually we need to collect the family-based study, getting the individuals from the family so that can be more concentrated and to study this. But the next generation sequencing, if we design our game right, and we can look at the entire spectrum of this. So the limitation of the GWAS, the first one is that they do not capture rare variants. They're simply not measured on these SNP arrays. And the second one is they usually do not represent the real functional information, meaning the SNPs you identified. If you think, I identified this SNP with this RS number, so let's see what this SNP does. Usually that is the wrong route to go. And those SNPs are normally only the tag SNPs, which means they represent a longer region in the genome, and the real causal SNP is within that region. So there will be a lot more informatics need to be worked out to understand the functions of the SNPs being identified. And there is also a lot of other type of heritability being missed because of the gene-gene interaction, gene-environment interaction, and also the epigenetic effects and other type of variants. So those are in general the limitations of GWAS, and I want you to understand this from conceptual level. And we also talk about the clinical application of this. And I gave two examples. The first one is a report in the clinic, and the clinical report paired with this individual genomic information and where we want to, what type of information we can infer and we want to infer in the future. And also there is a little kid that his disease was unknown and used the whole exome sequencing, and we found the gene and then eventually find the cure. And the last part in the previous lecture, we talked about the experimental considerations and about the DNA sequencing. And in terms of scope, there are several different scopes. The first one is whole genome sequencing, and it's getting cheaper and cheaper. And now it's probably close to $1,000 per genome. But and in a couple of years, it probably go to $700, $500, and ultimately $100 per genome. But the idea is that those probably is for research base. If we want to do anything at a clinical setting, and the cost is still pretty high. And I think for the clinical whole genome sequencing now, it still require about $4,000 at least of that scale. So, but I think in the next lecture, we're going to invite a clinician and come here to give a talk. Dr. Matteo Vata will be here and discuss about using different level of sequencing into the clinical setting. So when whole genome sequencing is becoming too expensive, and we can look at the targeted sequencing, and we really went through how do we select the target regions. And in most cases, that will be whole exome sequencing. And sometimes you extend that a little bit, go into the promoter regions or enhancer regions, or in many cases, through UTRs. And then to do that's the region selection part. And we also mentioned about the capture technologies. And in particular, we talk about the capture array and capture in solution. So if you forget about this and you want to go back to take a look at the notes. And if it's still too expensive to do this, and some people will do pooled sequencing to use a limited resource on a larger amount of populations. Of course, the biological question will be different. There will be caveat associated to those, but you pay what you got. And then we also talk about the long-range sequencing because we need to first get very accurate, and number two is we want to sometimes face the variants, and two variants, if they are nearby, in the nearby regions, we want to know who is coming with what. So in that case, we can either do long-range sequencing like the PacBio and Nanopore, or we can use something like a 10x genomics. They try to barcode a longer molecule and then sequence and then do a local assembly. So that will become much easier assembly approach, and that will increase your accuracy dramatically. And then we talk a little bit on the single cell DNA sequencing, just giving you a concept and present to you what are the real technological difficulties of those. And this is still a very, very active research area, and there is still very fast evolving. So in many cases, this year and next year, the technology could be extremely different, okay? So what we're going to talk about today is really getting into the informatics and statistical component of the DNA sequencing part. And so you remember that if I have to put an NGS into a central dogma, this will be my central dogma. We start from raw data, which is the FASTQ file, and then do the sequence alignment that derive the BAMS file, and then do the variant call and derive the VCF files. I mean, during this whole process, there are several different parts that I want to mention today, how people really do it. So the first one is in terms of the sequence alignment part. And I'm going to briefly talk about the concept of the sequence mappability, and also dig in a little bit on the refined alignment. So these two sections. And for the raw data and the VCF files, and I'm going to spend some time talking about this quality recalibration. Remember, at each of these three steps, we do have a quality associated to it, right? Base quality, mapping quality, and consensus quality. But these qualities can be recalibrated to get better performance. And then I'm going to discuss in the variant call perspective, and how do we do the variant call? What are the statistical considerations behind it? You might find that it can be really, really complicated. If you are kind of allergic to math, and that's the part you are going to have a lot of yawns. So we'll start from the first part, which is sequence mappability. And so if we revisit the human genome, for one sequence fragment, what is the minimum length can be uniquely mapped back to the human genome? So who can answer this question without looking at the notes? Do we know? Any biologists are sitting in the room did a PCR before? Do you design primers? How long are your primers? 20, 22, it depends on your temperature or the regions, right? And have you seen any primers that is 10 base long? Why not? Because it's going to be mapped on different locations in the genome, right? So if we do this math, we think of this, answer this question, if we think the size of the genome is about 3 billion letters, and then each nucleotide has 4 choices. So if we do this calculation, full power n is the number of the nucleotide in the either primer or our sequences, we need this number to be larger than 3 billion. So in that case, n is 15.7. So which means in minimum sense that your primer, sequencing primer, shouldn't be lower than 16 bases. Right? So if it's lower than 16 base, the chances you're going to find multiple spots in the genome. And so that is the real consideration on this part. So in theory, reads with 16 base or more can be uniquely mapped back to the human genome. Yeah, that's the theory part, which we know it will never work. And the reason for that is human genome are very repetitive. And actually there's a statistics, it's about 52% of the human genome is repetitive DNA, okay? And repetitive DNA elements are found in multiple copies, many, many different copies, thousands of copies in some cases. in some cases, thousands of copies in the human genome. And there are many different types of repeats. That will include something called tandem repeats, and we mentioned the last time, like mini satellites and microsatellites. Those are just, probably 2 to 6 base nucleotides and just repeat again and again and again for a long time. Or there's interspersed repeats, and that will include the DNA transposons, the long terminal repeats, LTRs, and retrotransposons, and the non-LTR repeats, signs and lines, and those type of repeats. So those are, in many cases, we consider that as junk DNA regions. They're just being in the DNA and the evolutionarily. I'm talking about not individual people when you're a baby until grow up. I'm not even talking about 10,000 years ago when we have an ancient human become today. It is talking about from lower organism and all the way evolved to human. And during the entire evolution process, and many things happened. And there are many things that somehow got into our genome, and sometimes multiple copies, and somehow that people have a way to block them not to use those regions, and sometimes we learn how to use those regions. So, but many of those regions are repetitive elements. Okay, so when we talk about this, so in very, very early phase, if we talk about back in 2007, 2006, the first version of Illumina at that time was called the Solaxa sequencers. Any of you know how long the base pair, what's the read length is? June, do you know that? Very early, it's not that early, it's 10 years ago, right? It's 25 bases, okay. And so in that case, actually, we know that in most of cases, we can uniquely map back to the genome, but we cannot guarantee how much we really map. So people actually, based on the human genome or different organisms in the genome, trying to calculate the mappability score. So X here is the length of the sequence. It's anywhere from 0 to 100 bases. And the Y here is the percentage of the genome that can be uniquely mapped by the length of, for example, 50. If we look at this, I don't remember which one is the human. Human is this blue one. So this line is human. And so if you look at 50 and you see here, and if that means if our sequencing rate is a 50 base pair, and that will indicate that 80% of the human genome can be uniquely mapped. Okay. But this is kind of contradict to what I just said. 52% of human genome are repetitive, right? In theory, we probably, you may think it's 48%. But actually, a lot of repetitive elements, they are not very long. There are a few dozen bases, sometimes a few hundred bases, and they are long ones, but in many cases are shorter ones. And they are kind of, just sitting in between some unique regions. So when you have a race, although one part is touching these repetitive regions, but the other part is still touching that unique regions. So although that part, even in those parts, they can be uniquely mapped, although they are technically repetitive elements. But you can see that from the human genome perspective, if we have the race that longer than 25 base, it's about 80% of the genome can be uniquely mapped. And further increase the mappability a little bit, but it's not dramatic, at least up to 100 bases. So, but this is something that you need to keep in mind of. Okay, and actually, it would be nice that we know that which part of the genome can be mapped uniquely, right? And actually, the mappability of uniqueness of the reference genome, it's kind of can be uniquely downloaded from the UCSC genome browser. Remember, I talked about UCSC genome browser in the very first lecture. But the general idea is in the UCSC genome browser, there are some tracks look like this. So you can see that these are the parts that can be uniquely mapped with 24 base all the way to 100 bases. And then there is the part that is not being mappable. So you can see that with the reads becoming longer and longer, at a certain point, that this gap will become smaller and smaller. So that will help you increase the read length, will potentially help you to do the mapping a little bit. But we know from the earlier slides, and it won't help dramatically, but at the individual size level, it still will help. Okay? So these are some of the concepts that you need to pay attention to, because it's very easy for us to generalize any results that we got from our data. And in many cases, we just plug in our data into some of software, it gives us the answer, and we believe in that, okay? In many cases, we are not that lucky, and it won't happen. And if something that looks too good to be true, normally it is too good to be true. It's not true, right? So when we find something that's, okay, I'm really ready now for my Nature paper, I identify something big. But now let's slow down a little bit and take a look at whether there are some potential technical artifacts. For example, when we identify genetic variants, so if there are two regions, they are very close to each other. I mean, they are very similar to each other. And then they have just one base differences, and both of them are homozygous. So if your alignment algorithm isn't done perfectly, and you might make a wrong conclusion, you might map this race back to this region, and then you are seeing, oh, there's a heterozygous variance. So sometimes we have seen this, and we have made that mistake before. And there was once that we work on a region that in the sex chromosome actually, and it's a female, and the X, we're supposed to see the heterozygous variance. And then we identify some heterozygous variants, and it looks really good, and none in the control, and there are some in the reference. But eventually we figured out there is a pseudo gene that is very, very similar, that is close by, that we are identifying artifacts. So really just be very careful on this type of things. So this will be the wrong conclusion. And be extra precaution when you really identify these variants. But sometimes when we look at the RNA sequencing experiment, we may potentially have this as well. So you can see that this particular two genes, their expression level is similar. But if there is another gene that is pretty close to this, and then you have some of the rates being identified here, if you're not paying attention to this mappability issue, and it's possible that you made the wrong conclusion, map all these rates back to here, and then you will look at it to say, okay, they're doubled. Actually, they are not. And there's a copy number variation potential issues as well, and there will be very, very similar that will lead you to make the same mistake. These are something that it's just the people do not pay attention to in many cases. And also that in the earlier days, when we don't really have the company design the whole exome sequencing probe for us, and we have to design that, and sometimes we need to be careful as well, because let's say this is a target region you want to do the sequencing, and then you say, I'm going to design so many different probes to target this target region. Nothing wrong with it, that's perfect. But when you use the PCR-based capture, when you do the analysis, you need to make sure that in the target region, there is no repetitive elements that is kind of repeat with each other. And sometimes it could happen. But especially when you look at this hybridization-based capture, like we talk about capture array or capture in solution. And in some cases, we not only need to pay attention to the within this target area mappability, whether they map with each other. And sometimes we need to look for whether this region is going to map outside, because if it can be you design a probe to target here, and actually it may also grab some part that's outside of the target region down to do the sequencing. And that will lead you to make wrong conclusions because you're going to see many genetic variants in those regions. So these are the things that we need to pay attention to when we do this type of analysis, and just be careful about that. And my probability is determined by many different factors, including the alignment algorithm you are using, and then including the regions where mapping occurs, and including the biochemistry assays. Sometimes maybe one technology to do the capture, for example, or even do the sequencing has a different sensitivity in terms of the mapping potential compared to different regions. Okay, and many of these actually you can generate this type of track by yourself by doing a certain level of simulation if you need to. Okay, now we don't really get into that deep into making sure everything is correct, but just be careful that pay attention to have something that in your mind that there are mappability needs to be considered. Okay, any question? All right. Now, the second topic about statistics about bioinformatics in DNA sequencing will be refined alignment. Okay, so let's see what that is about. Now let's first revisit our alignment algorithm that we talked about in the past. And so if you look at the sequence alignment, one thing, regardless of which algorithm you use or whatever method that you try to get the job done, but for the alignment, you are really trying to say that what you try to minimize is a number of mismatch bases minimized across one sequencing rate. So let's say this is your reference genome, and this is your sequence that you just got from a donor. And what you want to do is to make sure that the number of mismatches is minimum within this one sequencing rate. Does that make sense? So whatever is dynamic programming or whatever you use. And but the sequence for the refined alignment, and there's a difference. If this occurs in exam that you may need to answer this type of question is the number for the refined alignment, the number of mismatch bases will be minimized across all the sequencing rates. So we'll get into specifics what that really means. Okay, so let's take a look at what went wrong for the initial alignment. So a large percentage of regions requiring local alignment are due to the presence of insertion or deletion in the individual's genome with respect to the reference genome. So now let's go through this figure. This is very, very important. Okay, so I'll turn this around a little bit so that this can hear me for the recording purpose. So let's use this panel so you can see that here is the reference genome, okay? And the second one labeled as a sample. This is the real genome that you sequence for this particular patient, okay? And the bottom ones are the sequencing rates or individual sequencing rates. And so just keep in mind, so what is visible to you is the reference genome and all the rates. The sample, this particular row, the second row, these sequences are not visible to you. That is exactly something you want to identify, right? So you only have the reference genome, and then you have all these sequencing rates. You want to, by the end of the day, refer this reference, the sample genome, this particular donor. Does that make sense? Okay, so, but let's look at this. If we already know the answer, and you probably will see that there is an AAAT insertion in the genome you want to sequence compared to the reference genome, okay, in this case. So now let's see all your reads alignment. So what is being aligned here? They are all wrong alignment, okay? So you can see the first read, everything looked good until here. So you see this is the AAATAA. So this is the this should be AAATAA, right? But because we don't have this visible, so there's no way we can guess that. And then what people do is we'll align to here, AAATAA, with the consideration this particular A is a sequencing error. That makes sense? There's nothing wrong with the initial alignment. The initial alignment is aligning this read to the reference genome. If you align in the correct way, and you will see there are 4 nucleotide differences. And if you align in this way, you are seeing just 1 reference nucleotide difference. So this is minimizing the number of mismatches for this particular read compared to the reference genome. The original alignment did perfectly, it's just wrong, okay? And look at the second read, and it's supposed to be AAAT, AATT, AAAT, AATT. And now you don't do this, but rather you align here and consider this A as a sequencing error, this T as a sequencing error. It's still only 2 mismatches. If you do it right away, it's 4 mismatches, okay? And then let's look at this side. It's the same thing. That this is the CAAT. And then if you put this CAAT here, and it's actually CAAT. And it should be perfect match. But they will have to adjust it to here, and with the consideration, there are 2 mismatches. Okay, so the only read they aligned correctly is read number 3. So you see that this end and this end, they all have a significant span across this indel region. And they say, okay, this read aligned to this has 4 nucleotide differences. And that is the lowest, the smallest I can ever find. Okay? So you can see that the original alignment didn't do anything wrong. They were trying to minimize the number of mismatches comparing to the read versus reference genome. But because the real correct answer is not visible, so there's nothing they can do at that stage. So such alignment artifacts result in many base mismatching. So like this, you see very sporadically some of the mismatches around this area, mismatching the reference near the misalignment, which are mistaken or as SNPs. Does that make sense? So that is, first of all, before we try to find a way to solve this problem, and let's first understand what the problem is. So here is the problem, okay? And the indel mapping treats each read independently, so they are not really looking the whole picture together. So even when some of the reads are correctly mapped in indels, reads covering the indel near just the start and end are often misaligned. So you can see that. So local alignment is precisely trying to solve this problem. And how to do that? There are 2 steps. So the first step is to identify the regions that are misaligned. And the second step is to fix it, okay? So now let's see how to do the identification, how to do the fixing. So the identification is you will first need to detect suspicious regions. Which region is easier to get this misalignment? So there are several different criteria. The first one is de novo indels in the initial alignment. So meaning that in the previous case, remember we have a read 3 that has the indel being correctly aligned. So if you identified some hints in your data, there is an indel, that is a suspicious region we need to go back to take a look at. And the second one is if there's no indel being identified in those particular region, sometimes you may overlook it. But if you already did the base calling, and some of the regions somehow you just feel it's not right, you have identified many different SNPs across a very close area, that is the indication there may be need to have refined alignment in those regions. But sometimes when you haven't do the base call, meaning you already did the alignment but haven't called the SNPs, but you see some of the regions that there's a lot of inconsistency across the same site but with the nucleotide, and which they mean this high entropy, and then that region is kind of suspicious. And also that you want to look at the known indel regions in the DB SNP. So those are the first step to detect the regions that is possibly requires refined alignment. And the second step is to fix it. And to fix it, there are several different iterations. So the first one is you want to construct all the possible haplotypes by integrating the reference genome, the known gap, and the de novo gaps. So what this really means, okay? So what this means is you have, initially you have your reference genome. That is only the reference genome. Now you identify some suspicious region, right? And then now let's make another reference genome. So meaning that if we go back to this step, is initially we only have this one reference genome. That is our original haplotype. But because of this read number 3, have an indication there are something going on as AAAT, as an indel insertion here. So maybe there needs to update our reference genome. So you have a second reference genome by inserting this AAAT into your original one. So now you have 2 reference genomes in a very localized area, only in the suspicious area you identified. That makes sense? So that is called 2 type of multiple haplotypes. So you want to construct all the possible haplotypes by integrating this information together. And then what you do next is to do a gapless alignment, meaning that this next step, when we do the alignment, we do not allow insertion deletions. But when we do the alignment, we want to align all the reads to the original haplotype reference and the new reference I just built, okay? Now let me go back to this again. I have this one, I have this one, I have 2 haplotypes. And then for every single read that in this area, I'm going to let it align to here and then align to here. So I have 2 different alignments based on 2 references that I just built for this region. At this stage, I do not allow indels, just the perfect, needs to be perfectly aligned. So if you look at this, if we do this for this particular region, for read number 1, when this one is being aligned to here, and it's still this, right? But when this one is being aligned to the second haplotype, now we know this AAATAA will all be aligned to the right spot. Okay, so you do this for every single read that is in these regions. Okay, and so it's like this. So you have your reference, you have your new reference haplotype, and then you map all these reads to both of them. Okay, and then we are having a little bit math here, and this will be the equation that you are used to calculate how likely the new haplotype is a real haplotype. So there is a real insertion deletion. So let's, we will have some quiz or exam questions about these equations. No, we will not, but let's still try to figure this out. I mean, to give you a general feeling that what this really means. First of all, this LHI indicates the number of haplotypes. Okay, so for each haplotype, ultimately we want to calculate the likelihood of this particular haplotype. Okay, and this is being determined by this equation, this part, which there's a number of reads is J in this region. So let's say we have 10 reads here, and then there will be 10 things timed together. And then each one is following this equation, which is here actually. And the way to do that is meaning this really means if this is the right haplotype, if this is the right reference, what's the likelihood I'm going to observe this particular read, right? Remember when we have the read and there's a mismatches, for those mismatching locations, we have the quality score. So we already know what's the probability that we make a mistake. So this one, L, this number, and the K will be the number of mismatches here. And then you can see that, each individual one will be calculated by this. So basically just to say, if this is the right haplotype, I'm identifying 2 SNPs in these regions. If this is the right haplotype, I'm identifying 5 SNPs in these regions. Okay, these SNPs, they are all sequencing errors. How much possibility I'm really making a mistake here, okay? So by using this, you basically have a fair evaluation whether this is the haplotype or this is a haplotype for this particular region. Remember, we are diploid genome, so meaning that we have one haplotype from father, one from mother, so it's an opportunity that both of them are correct because your parents may not agree with each other on their genome, the same as everything else. But the bottom line is you may be a hybrid of both. So when we sequence your sequencing rates, it may have a possibility both 2 haplotypes occur. So basically you calculate the likelihood of your new haplotype, okay, for those who don't worry about that. And then we want to, the haplotype HI that minimize this is selected as the best alternative haplotype. Okay, so our reference will always be the haplotype, but this is a best alternative haplotype here. And there is a further equations I'm not going to go through, which means that if my new haplotype, the score, the likelihood score is only a little bit better than the original one, I'm not going to take it. It has to be way better than it. Actually, usually they have a threshold like a 5 or something. Did I write it somewhere? The rates are realigned if log odds ratio of the 2 haplotype model is better than the single reference-type at least 5 log units. That is very, very significant. So meaning that I'm going to evaluate both of them. And if adding a new option will be resulting in much way better results, I'm going to take it, okay? But you can go back to take a look at the entire logic of this section. It's not that complicated. And then we are not going to do the exam on the match part of it, okay? But let me show you a few figures about the performance. So you can see that this is in their Nature Genetics published. This is everything, most of the stuff I talk about today is coming from the GATK package. That is what Sean is going to teach you how to run. I don't remember, probably 2 lectures from now. And that is so important. And it's kind of the gold standard for the DNA sequencing analysis. So but you can see that in this case, there is 3 SNPs here. And actually in this region, this is suspicious because some of the reads identify the indel, okay? After they go through the math that I just presented, they actually have a way to fix this region into this. So you can see this is actually a homozygous, a heterozygous variant, but the variant type is an insertion deletion. Actually, you can see this part is not colorful anymore. And this is another example. So you can see there's indel regions, and there are more variants that we really usually see. And then after this, actually you are seeing here is almost a homozygous indel regions. So in this case, that person's parents actually agreed with each other in terms of their genotypes. Any questions so far? Okay, so just key information summary on this part is, so this is the take-home message for refined alignment. And the first one is, when we do the refined alignment, we are not trying to minimize the mismatches of one read. We are trying to minimize the number of mismatches across all the reads in this particular region. That is our goal. And there are two-step process. The first one is we need to identify suspicious regions. The second one is really to do it. And to do it, there are three steps. The first one is to enumerate potential haplotype candidates, and then do the gapless alignment on all the candidates, and then calculate the likelihood for each haplotype. Eventually, we make the decision whether we decide to take it or not. Okay, so, but we need to also, there's another thing we need to pay attention to is, this is something that we do for the most DNA sequencing analysis, but we need to, after you know this, if you think of a little deeper, there are sometimes certain scenarios we cannot use the refined alignment on DNA sequence. So the major assumption here is this algorithm operates on all reads across all individuals simultaneously, which ensures consistency in the inferred haplotype among all the individuals, which is great, okay? But this doesn't work on somatic SNPs, okay? So if you're working on the cancer genome, if you are working on something that, that only a small percentage of cell has that SNP, and you might see a small percentage of cell has that SNP. And when you do the refined alignment, even there is a small insertion deletions, it may not be able to give you enough advantage by bringing in a new haplotype mathematically. Remember, we need that 5 log unit of the advantage. So if you only have a very small percentage of the cells in the cancer tissue have that small indel, you may not be able to identify it, okay? And the need to have significant input to replace the initial alignment may not work for pooled sequencing, because if you pool 10 people's DNA together, maybe only one person's one allele has that indel, and then you are talking about only 5% of the other reads for the indel. So again, you may not gain enough advantage of that. RNA sequencing, sometimes we don't do it because first of all, usually we do not call indels from RNA sequencing, but also we need to be careful for the RNA sequencing. 2 alleles may not express at the same level as well. One allele may express more than the other ones. Okay? So that is the refined alignment part. So let's take a break for 5 minutes. We're coming back 3:55. Okay?